A'udhu billahi minash shaitanir rajim. Bismillahir rahmanir rahim. Assalamu alaikum wa rahmatullah. This is part 2 of my research work. In part 1, I explained about foot and mouth disease and its pathogenesis, treatment, clinical signs, and my objectives of my research, and then methodology adopted to complete those two very important objectives. Now the results. Based on NSP ELISA in bovines. Here is a graphical representation which indicated that the tehsil-wise distribution of occurrence of foot and mouth disease virus in bovine population based on NSP ELISA. That indicated that highest seroprevalence was noted in tehsil Ahmadpur East, followed by tehsil Khairpur Tame Wali, and Bahawalpur, Hasilpur, and the lowest seroprevalence was noted in Yazman, which indicated that these two tehsils, Ahmadpur tehsil and Khairpur Tame Wali, they were highly infected with the FMD virus, and that may be due to the fact that the animals which are living in these two tehsils, they are living under less hygienic conditions and less biosecurity conditions, and movements of these animals are not well restricted. So because if movements are restricted, there are less chances of the infection. Fig 2 indicated tehsil-wise occurrence of foot and mouth disease infected cows and buffalo population based on NSP ELISA. That also indicated that highest seroprevalence was noted in buffaloes of tehsil Ahmadpur East, followed by the buffaloes of tehsil Khairpur Tame Wali. In tehsil Bahawalpur, cows population was showing higher seroprevalence, means infection rate is higher in tehsil Bahawalpur in tehsil Bahawalpur, in the cows of tehsil Bahawalpur, followed by buffaloes of tehsil Bahawalpur. In Yazman, we can see that buffaloes were showing higher seroprevalence as compared to the cows. In Yazman, cows population infection rate is at 0 level. In Hasilpur, infection rate is at higher level in cows. So overall, we can see that tehsil Ahmadpur East and tehsil Khairpur Tame Wali, these two tehsils, animals were at higher risk, and specifically buffaloes population were at higher risk. Here is gender-wise occurrence of FMD infected cows and buffaloes based on NSP ELISA. So we can see here in graph that blue color indicated bulls and cows. Blue color indicated bulls here in male portion and cows here in female portion. Seroprevalence or occurrence of FMD infection was noted higher in female cows as compared to the male cows, and pattern is the same. Occurrence of FMD infection rate is higher in case of buffaloes, female buffaloes, as compared to the male buffaloes. And reason behind this may be that females are kept for longer period of time, number one. Number two, they suffer from so many stresses like pregnancy, that is a great stress on them, and similarly heat periods and so many others. These stresses may lead to less immunity in females and more exposure to the FMD virus. Here is table 1, age-wise distribution of FMD in bovine based on NSP ELISA. That indicated that in bovine calves and among bovine calves and bovine adults, we can see that with the increase of the age, prevalence was also increasing mostly, and Among bovine calves and adults, higher seroprevalence or infection rate was noted in adults. And adults having more than 4 years of age, they were showing higher seroprevalence, that was 20.5%, followed by the animals having 2 to 4 years of age. And among bovine calves, 7 to 12 months of aged animals were showing higher seroprevalence, followed by the 3 to 6 months of age. This is tehsil-wise seroprevalence of serotype Asia1, O and A of FMD virus in bovine based on solid phase competitive ELISA seroprevalence 2 study results are now being discussed, that indicated that Tehsil Ahmadpur East was showing higher seroprevalence, followed by Tehsil Khairpur Tamewali, then Tehsil Bahawalpur and Hasilpur, and finally Yazman. And among 3 serotypes, serotype O was noted at higher level. Highest seroprevalence, highest rate of infection was noted of serotype O in Tehsil Ahmadpur East, followed by Tehsil Bahawalpur of serotype O. Here trend is somewhat different. In these 3 tehsils, serotype O was at higher level, followed by serotype Asia1 and serotype A. But in Tehsil Hasilpur and Khairpur Tamewali, trend is somewhat different. Serotype Asia1 was at the higher level. Prevalence of serotype Asia1 was noted higher, followed by serotype O and A. Here is overall occurrence or seroprevalence of all 3 serotypes in bovines in whole district, that indicated that serotype O was found 28.5%. Prevalence level is 28.5%, followed by serotype Asia1, which is 23.6%, and finally serotype A, that is 13.6%, and the p-value indicated that statistically significant prevalence was found statistically significant. Tehsil-wise seroprevalence of serotype Asia1, O and A in cows population indicated that Tehsil Ahmadpur was found at higher level infection, and serotype O was the serotype which was showing the highest seroprevalence, followed by serotype Asia1 and serotype A, and then Tehsil Khairpur Tamewali. In cows, these 2 tehsils again they were highly infected. In these 2 tehsils, cows were highly infected, and then this is tehsil-wise occurrence of serotype Asia1, O and A in buffalo population, that indicated that Ahmadpur East tehsil was showing higher seroprevalence of serotype O, followed by Asia1 and A. And then Khairpur Tamewali, here Hasilpur is at minimal level. Tehsil Hasilpur buffaloes population were less infected as compared to the all other tehsils, means are showing that the animals, buffalo population especially, they are at lower risk level, and infection rate is very less in these buffalo populations. Here is age-wise occurrence of FMD serotypes in bovine calves population of district Bahawalpur. There are 3 groups of bovine calves: less than 3 months of age group, 3 to 6 months of age group, 7 to 12 months of age group. That indicated that the highest seroprevalence among bovine calves was noted of serotype O, that was 32.4%, followed by serotype Asia1, that was 29.7% in the same age group. And then comes the serotype 3 to 6 months of age group of serotype O. Prevalence was noted higher of serotype O overall in this age group. Then comes the age-wise occurrence of FMD serotypes in bovine adults population of district Bahawalpur, and that also indicated that highest seroprevalence was noted in more than 4 months, more than 4 years of age animals, and serotype O was at highest level, that was 38.4% in more than 4 years of aged group animals, followed by 2 to 4 months of age group animals. This is overall distribution of foot and mouth disease serotypes Asia1, O and A in vaccinated and non-vaccinated bovines in district Bahawalpur. This study indicated that serotype O was found at highest level, higher level. Prevalence of serotype O was higher in vaccinated and also in non-vaccinated group of animals of bovines, followed by serotype Asia1 and serotype A. Overall, we can say that serotype O was noted at higher level. Infection rate in animals was of serotype O. This is gender-wise, gender-wise seroprevalence of serotype Asia1, O and A of FMD virus distributed among bovines, that indicated that female cows were showing higher seroprevalence than male cows, and female buffaloes were showing higher seroprevalence than male buffaloes. And among all these 4 groups, female cows were showing higher seroprevalence. And if we see that among 3 serotypes, serotype O was showing higher seroprevalence. Molecular identification of foot and mouth disease virus in bovine, that indicated that serotype O was identified from the animal cow of Tehsil Ahmedpur East, and accession number was obtained from NCBI, accession number OK359042, that showed 62% identity with FMD type O, previously reported in the cattle of Janwala Sargodha, Pakistan. And serotype O that was identified in our samples from the buffalo sample taken from the Tehsil Khairpur Tamewali, accession number was obtained OK359043, that showed 71% identity with FMD type O, that was previously reported in the cattle of Janwala Sargodha, Pakistan. This graphical representation indicated Tehsil-wise distribution of FMD virus seroprevalence in domesticated animals based on NSP ELISA. Okay, so that indicated that the highest seroprevalence, highest rate of infection was noted in Tehsil Ahmedpur East, followed by Tehsil Bahawalpur, Yazman, Hasilpur and Khairpur Tamewali. Species-wise overall seroprevalence in the whole district based on NSP ELISA indicated that highest seroprevalence was noted in goats, followed by sheep, and then finally camel. So role of the camel should not be ignored, and similarly role of the sheep and goats, that should also not be ignored, and immunization process should be continued in these. Here we can see the gender-wise seroprevalence of foot and mouth disease virus distribution among sheep, goats and camels, that indicated that highest seroprevalence was noted in the male goats as compared to the female goats over here. But in case of sheep, highest level of seroprevalence was noted in females as compared to the males. And in females of the camels, 4% seroprevalence was noted, but in males of the camels, 0 seroprevalence was noted. This is age-wise distribution of FMD in domesticated animals in NSP ELISA. Among calves and adults, if we compare, this is very evident that highest level of seroprevalence was noted in adults of domesticated animals, and among adults more than 4 years of aged animals, they were showing 20% seroprevalence. Fig 13 indicated tehsil-wise seroprevalence of all three serotypes in domesticated animals, and it is very evident from here that tehsil hasilpur of domesticated animals was showing highest rate of infection. So, and these animals of domesticated animals of tehsil hasilpur, they were highly infected with serotype O, that was 46.1%, and serotype O in Yazman was 39.7%. So level of the occurrence of serotype O, that was found highest in Hasilpur, then in Yazman, and after that Khairpur Tame Wali. Lowest level was found in Ahmedpur East. Serotype O was found at lowest level in Ahmedpur East. Here is the species-wise occurrence or seroprevalence of all three serotypes of FMD virus in domesticated animals, that indicated that in goats, serotype O was found at 44.5% seroprevalence level, followed by serotype Asia 1 and A. Then comes sheep, and finally the camels. Level of seroprevalence was noted 0 in dogs, cats, and donkeys. Here we can see the graph of the seroprevalence among domesticated animals age-wise distribution. Age-wise distribution indicated that highest level of occurrence of serotype O was noted in 7 to 12 months of aged animals, followed by 2 to 4 years of aged animals in domesticated animals. And we are very clear from here that in domesticated animals we included mostly sheep and goats and then camels. So sheep and goats, they are kept mostly for 2 to 4 years of age animals, and then after few animals are kept for more than 4 years of aged animals. So here trend is that the 7 to 12 months of aged animals, they are suffering highly, they are infected highly with the serotype O followed by serotype A. Gender-wise seroprevalence indicated that female goats were showing higher seroprevalence of serotype O, followed by male goats showing 40.1% seroprevalence. So overall seroprevalence O was noted at higher level in female goats, in female sheep as well, and also but not in female camel. In female camel, serotype Asia 1 was noted at 8% level. Here are only few slides which are remaining, 4 to 5 slides only. Phylogenetic analysis of FMD virus conducted through partial sequence of VP1 gene, that indicated that 2 samples were positive. One was positive from the sheep of the Khairpur Tame Wali, and the other was positive from the goat of Khairpur Tame Wali. Accession number was obtained, and that they were OK359044, and this accession number serotype indicated 67% identity with FMD type O, that was previously isolated from the cattle of Iran. And other accession number that was obtained from the sample goat of Khairpur Tame Wali indicated 63% identity with the FMD type O, previously reported from the cattle of Khanewal, Punjab, Pakistan. Molecular identification of FMD virus from domesticated animals indicated the same results as I have described in this slide, that 67% similarity, and here is 63% similarity of that serotype O, which we identified from the sample of sheep and goat from the Tehsil Khairpur Tame Wali. This is phylogenetic analysis of FMD virus through partial sequence of VP1 gene that indicated the location of these accession numbers. Accession number 42 OK 359042, OK 359043, OK 359044 and 45. This is the location. Conclusion of our study is that the present studies signified the dynamics of prevailing FMD virus and its serotypes circulating in the district. And we also concluded that serotype O was found at higher level and cows were the population that was at more risk level. So cows were found more susceptible from serotype O, and goats were the animals or populations that was found more affected from serotype O, and overall the whole district was found at endemic level, means that there is regular continuous occurrence of the disease. Role of the camel and sheep and goat should not be ignored. Immunization process should be continued, at least in sheep and goat as well, which is not being followed nowadays. Only cows and buffaloes, they are being inoculated. So there is recommendation that in recommendation portion we can see that we can add this, the population of the sheep and goat, they should be inoculated. And we also concluded that 62 to 71% identity was shown by these two serotypes with the previously reported serotypes of Sargodha, and OK 359044 and 9045 indicated 67 to 63% identity from the cows of Iran. Pattern of seroprevalence of serotypes of FMD in vaccinated and non-vaccinated bovines was same, indicating that present vaccine of FMD have equally good coverage of FMD serotypes which are prevailing in non-vaccinated population. So age, species, gender, area, like tehsils, they remain the important risk factors in our study, and findings of our study, they will help in strengthening the FMD control program in Pakistan to develop FMD free zones. Future recommendations, the 4 different serotypes of FMD virus type O, OK 359042, OK 359043 and 44 and 45, they may also be incorporated in future vaccination program, and they should be incorporated so that the serotypes which we identified, they should also be covered in immunization program, and there should be no risk from those serotypes. Regular inoculation program with trivalent vaccines in bovines, sheep and goats should also be incorporated. Role of camel should not be ignored. As mentioned earlier, proper vaccination monitoring of inoculation programs, effective biosecurity measures, capacity building of veterinary staff, workshops on FMD control programs and sample collections, they should be arranged. Strengthening of veterinary diagnostic labs, that is necessary. Restricting the movements of animals to prevent more exposure of the animal, and infected carcasses should be disposed of properly, like incineration and burial techniques. Here are a few references from where we took the whole material. Here is our article that has been published, Alhamdulillah Rabbil Alameen, in Sarhad Journal of Agriculture, volume 39, issue 2, page 308 to 315. Title of our research work, title of our article was Circulation of Predominantly Prevalent Serotypes of Foot and Mouth Disease Virus in Bovine Animals in Bahawalpur, Pakistan. We acknowledge all professors, including in our supervisory committee. We also acknowledge the Dean Faculty, Professor Dr. Farzana Rizvi, We acknowledge Dr. Muhammad Afzal, FAO expert on FMD, who initiated FMD control plan program in Pakistan. And we also acknowledge lab staff and colleagues, also acknowledge Dr. Muhammad Shahid Mahmood, and we acknowledge NVL Islamabad, LNDD Department, Secretary Livestock, Hotiana Sahab, and Dr. Muhammad Siptan Bukhari, Director Livestock Division Bahawalpur. And we also acknowledge the whole veterinary staff of District Bahawalpur, especially CVH Chenigot. Thank you very much. Jazakallah khaira. Thanks for patience.